Good morning. Let's discuss on urea cycle. The cycle is also known as Krebs-Hensley cycle, and the cycle is also known as ornithine cycle, as the first of reaction is ornithine. So the reaction is a reaction of the urea cycle are happening into the liver hepatocyte in both the compartment mitochondria as well as the cytoplasm. Initial two reaction steps will happen to the mitochondria, while the remaining three steps will take place in the cytoplasm of the hepatocyte. So carbon dioxide we require for the urea synthesis. It comes from the Krebs cycle, which is also happening in the same compartment. Plus ammonia is being derived from the oxidative deamination of glutamate, and both of these combined or condense in the presence of enzyme carbamoyl phosphate synthetase 1 into the form of carbamoyl phosphate. And in this reaction is being activated by the N-acetyl glutamate. N-acetyl glutamate or NAG is the alloesteric activator of this enzyme, carbamoyl phosphate synthetase. Further, carbamoyl phosphate is converted up into the citrulline in the presence of ornithine, and this ornithine comes from right from the cytoplasm via the ornithine transporter. And this ornithine in the cytoplasm condensed with the carbamoyl phosphate in the presence of enzyme ornithine transcarbamylase. Citrulline once formed, it shifted to the cytoplasm. In the cytoplasm, citrulline contains with aspartate, leading to the formation of arginosuccinate. And here the two ATPs are uh, one ATP is broken up into the AMP. So there are two high energy bonds are broken up, which are equivalent to the consumption of two ATPs. Then further in the step 4, the arginosuccinate in the presence of arginosuccinase is broken up into two pieces. One is the arginine and another one is the fumarate. And fumarate goes and utilized for the citric acid synthesis. Then arginine further converts up into ornithine in step 5 with the release of urea. So step 5 leads to the final release of urea, and the enzyme here is the arginase. So there is a total compartmentalization of the 5 steps to initial steps, and the urea is being generated in the cytoplasm. So here I would like to explain about the bioenergetic also. Like here there is in step 1 consumption of 2 ATP plus the consumption of 2 ATPs here, because ATP is converting directly to AMP, so that is equivalent to for 2 ATP. So total 4 ATP are consumed here plus fumarate which is being generated. It goes to the citric acid cycle. In citric acid cycle, it generates 1 NADPH via converting to malate to oxaloacetate. So 1 NADH is being generated, which is equivalent to a generation of 2.5 ATP. So total loss of ATP in this particular reaction is 1.5 ATPs. So this is the summary. This is about the enzyme. Like the enzyme which we are using in step 1 is carbamoyl phosphate synthetase type 1. And there is another enzyme which is responsible for the pyrimidine synthesis in the cytosol, that is the CPS2. So what are the differences? Have a look, because site is of CPS1 is the mitochondria, while CPS2 is cytoplasm. Pathway, it is used in the pathway of urea cycle, and while it is in the pyrimidine synthesis. Positive effector is NAG of the CPS1, while there is no no positive effector or negative effector for the CPS2. Then sources of nitrogen is ammonia for CPS, and while for CPS2 the sources are glutamine. Then there is no inhibitor of CPS, while CPS2 there is a CTP as act as an inhibitor. Then this is a summary about the site is the liver. Arginase enzyme is only present in the liver. The first two reactions happen in the mitochondria. Next three steps happen in the cytoplasm. Total five important enzymes are contributing to the formation of urea. Then one activator, that is N-acetylglutamine, that is an alloesteric activator of CPS1. Then leading any enzyme defect of any of the five enzymes will lead to five disorders plus triple H syndrome. We will discuss about it. Then compartmentalization. What is the scope of compartmentalization? As urea cycle enzymes are located in such a way that first two enzymes are in the mitochondria and rest are in the cytoplasm. So the inhibitory effects of fumarate on its own formation is minimized because arginosuccinate lyase is in the cytoplasm while fumarase in the mitochondria. So about the significance about the urea cycle, toxic ammonia is converted into the harmless and non-toxic urea. Then ammonia and carbon dioxide, these are the two waste product of the body which are to be converted up into the non-toxic substance, that is urea. So this is how we are disposing of the wastage. Then it participates in the regulation of pH, which depends upon the ratio of dissolved carbon dioxide. So carbon dioxide in the form of carbonic acid or in the form of bicarbonate ion. So these are the major pH regulator or buffers of the blood. Then it forms the semi-essential amino acid, that is the arginine, and arginine also serve as the potent precursor for synthesis of nitric oxide, which is a potent muscle relaxant. Ornithine, which is formed in the urea cycle, is a precursor for the formation of polyamines like patricine, spermidine, spermine can also be involved in the formation of non-essential amino acid proline. So I have look on disorders associated with the urea cycle. There are five disorders and one syndrome is there: hyperammonemia type 1, type 2, citrullinemia, arginosuccinate aciduria, argininemia, and then triple H syndrome due to the defect in ornithine translocase enzyme gene. So let's discuss one by one. The hyperammonemia actually characterized by increased level of ammonia, which lead to encephalopathy and respiratory alkalosis. The clinical symptoms include the vomiting, irritability, lethargy, severe mental retardation. Infants appear normal at birth, but within days they show a lot of lethargy. The treatment is more or less similar to the similar in different types of disorders. Like we can suggest them to have low protein diet with sufficient arginine and energy by frequent feeding. um, uh, we can minimize the brain damage since ammonia level do not increase very high at a time. So, type 1 hyperammonemia, it is due to the deficiency of carbamoyl phosphate synthetase. Another variant of this disease is due to the deficiency of the coactivators, that is the NAG. So, it is also characterized by severe hyperammonemia of grade 1 because it is the just step which is consuming the ammonia. So, if it is lacking, so a lot of accumulation of ammonia and it is spreading, especially the brain faculties are very much, they are worstly affected, leading to the encephalopathy, and respiratory alkalosis is also there. Then second type of disorder is hyperammonemia type 2. So type 2 is the only urea cycle disorder which is inherited as an X-linked trait, and this is characterized by OTC deficiency. Elevated level of ammonia are associated with high glutamine level in the CSF and blood. High mitochondrial level of carbamoyl phosphate will allow its passage into the cytoplasm. It will move into the cytoplasm and it will divert to the synthesis of pyrimidine, and in course of that synthesis it will lead to the synthesis of high concentration of orotic acid, and that is going to be excreted through urine as well. So then when ammonia is increased, the brain synthesizes more glutamine for ammonia fixation, which is present in the CSF. And third type is the deficiency of arginosuccinate synthetase. So arginosuccinate synthetase deficiency is characterized by hyperammonemia and citrullinuria, because this citrulline is accumulated severely, so it starts coming into the urine as well, from blood to urine. The next type of deficiency is due to the deficiency of arginosuccinate lyase. It leads to the arginosuccinate aciduria, and therefore metabolic acidosis happens. And hyperammonemia is less severe, and arginosuccinate is elevated in CSF and is excreted in the urine. So a typical clinical feature is friable tufted hair. So this is the very important feature of this arginosuccinate lyase deficiency. The next is arginase deficiency, the deficiency of arginase. So it's a mild variety with accumulation and excretion of arginine. Symptoms appears only by 2 to 4 years of age. The accumulation of ammonia in the blood is less than 40 micrograms per dl, and even the small amount of metabolic intermediates accumulate in the blood of new born infant can be detected by the tandem mass spectrometry. So, and the next disorder is the triple H syndrome. That is actually due to the deficiency of the transporter, which is meant to carry this ornithine right from cytoplasm to the mitochondria. So this deficiency is characterized by hyperornithinemia, hyperammonemia, homocystinuria syndrome. Ornithine has to be transported into the mitochondria, and citrulline has to come out since the urea cycle is compartmentalized. So here what happens, the ornithine accumulates into the cytoplasm, and it's not, since it is not available into the mitochondria, so lysine is carbomylated to form the homocitrulline. So this is the overall picture of why there is triple S syndrome in defect of ornithine translocase gene. So that's all about the urea cycle. Thank you.